Okay, so we're back. Okay, I think maybe we'll start again, actually. My name is Stephen. I'm the community editor of the PloS Synbio blog. And we're here today to talk about, we're here today with Dr. Fuzhong Zhang and his group. And we're here to talk about the recently awarded grant, which was to investigate the production of lightweight materials in cyanobacteria. Before we get onto that, I just want to say thank you for taking the time to speak to us today. And I was wondering if you could give us a little bit idea about yourselves, maybe a quick summary of what you're working on at the moment.
Sure. So I'll start first. My name is Fuzhong Zhang. I'm an assistant professor at the Department of Energy, Environment, and Chemical Engineering in Washington University. My research focuses on synthetic biology and metabolic engineering. We are interested in developing synthetic biology platforms to make structurally defined chemicals and materials. We are also developing tools to make synthetic biology easier.
Okay.
So I'm Chris Bowen. I'm a third-year graduate student in Fuzhong, and started working with microbial production of monomers for plastic synthesis. And now I'm sort of doing the groundwork for this NASA project.
Cool.
Hi, I'm Eugene. I'm actually a first-year PhD student, so I actually just recently joined it, but I've kind of just been working on optimizing protocols and kind of the processes that have already been going on, and I'm really excited to be a bigger part of the project.
So how did you hear about the project? Eugene, yeah.
So I guess when I first applied to graduate school, I kind of looked into different labs, and Dr. Zhang's lab was of particular interest just because in undergrad, I had previous experience in research with synthetic biology and metabolic engineering, not necessarily with cyanobacteria, but the production of biomaterials was something I was really interested in. So yeah, this project was definitely something I wanted to be a part of.
Cool. So yeah, I mean, it's quite cool to say that you have a NASA grant. I was just wondering how you got into this. So I mean, I guess a lot of your research has been focused in kind of like gasoline production and things like that. But how did you come across this NASA grant, and what kind of problems were they looking to address?
Right. Probably since last year or somewhere last year, we started to move our focus from gasoline, from fuels to materials. And ever since then, we have been trying to solve one of the problems in renewable materials, which is how you can engineer a synthetic biology platform for the renewable production of these high-performance materials.
Mm-hmm.
So similar to the fuels that they have the same problems that their original feedstocks are running out, we need to look for alternative ways to produce them. And I believe that to solve these problems will require tools and techniques which are similar to those for the production of biofuels. Then we think that my group will have the right expertise to solve this problem. And almost for a little bit longer than a year, we have been working on this project. And later, we came across this opportunity from NASA, and we found that would be the right product for us because we have been developing these tools to make materials, and those materials could be the thing that will be needed by NASA. So I think, well, that's great. Then let's put a proposal, and fortunately, we were selected.
Okay. So Chris, is this something you've been working on then?
The NASA project?
No, the kind of material production.
Yeah, right. So I mean, that was my first project, is monomers for plastic synthesis. And this is kind of like an extension of that. Whereas with those, you have to purify those monomers and then do some sort of polymerization. In this project, we're just making the polymers directly, or the materials directly.
Cool. So yeah, I mean, that sounds very challenging. So I guess, was that in cyanobacteria that we were working with before, or was this, or was it
my work?
Yeah.
No, that was E. coli. And actually, right now, I'm doing this sort of groundwork in E. coli, and then I guess we move the project to cyano.
Okay.
So for cyanobacteria, maybe I can speak a little bit more on that. We do have a separate product that we have been working for almost 3 years now. That is one of the products that we started at the moment I started this lab, which is to engineer cyanobacteria to fix nitrogen. So again, that is a synthetic biology product. It involves with lots of new tools developed for cyanobacteria and how to control expression of multiple genes and how to control the behavior of large metabolic gene clusters. So we learn a lot for cyanobacteria, and at the moment when we are trying to address the issue for NASA, we think cyanobacteria will be the right host for that. So that's how we came up with the current product for NASA.
So is this on the idea of using like solar energy rather than as actually the power source rather than something which you have to feed in some, like, I guess yeast or E. coli, you have to feed in glucose or stuff like this?
Exactly. So that is one of the biggest advantage of using cyanobacteria, particularly for NASA applications, because I think the NASA's idea is trying to use this platform in a agricultural environment, or in NASA's word, in situ resource utilization. Where cyanobacteria will just play the right role because it can take sunlight, which presumably will be plenty, and it will take carbon dioxide, which could be either waste from human metabolism, or could be some plenty of gas resource that you can find from other planets. And at the same time, also cyanobacteria do have the potential, the metabolic potential, which you can engineer or you can manipulate for it to make certain products that that NASA has interest. And also, certain cyanobacteria species are fairly robust that they can thrive under a variety of extreme conditions. And for example, under high carbon dioxide pressure, or under some fairly low pressure environments. So I think all of these contribute to the fact that it would be the right host better than E. coli or Saccharomyces, those more traditional microbial hosts.
Okay, so I kind of a strange question, but like, is this going to be, well, is the ultimate aim would be it would be done at like zero gravity? So has anyone done any study of like how cyanobacteria are grown in space? Like, would it be in a bioreactor, or how would it happen?
Right. So first, I think there's one thing is either between we're going to use this, NASA is going to use this technology in space, doing space traveling, or in a extraterrestrial environment, which presumably will be not quite zero gravity. So I think that is NASA's decision and probably beyond our product. And in a way, to my understanding, NASA did spend time and effort to study how those photosynthetic organisms would grow under zero gravity conditions. So I think some of the challenges probably already been solved, and which is not the issue that we're going to address.
Okay.
So we're mostly focused on to engineer this host towards material production.
So what kind of materials you're looking to? Oh, sorry. Chris, would you be able to talk about what kind of materials you're looking to produce?
Yeah, sure. Basically, I mean, we're starting off with these lightweight, high-strength materials. And I guess part of the reason is because biology is already very good at making those materials, but also because they could be used for a lot of NASA applications. And we sort of envision applications like parachutes, or either making these materials or doing repairs on these items like parachutes or clothing or containers, that sort of stuff.
Okay, cool. So I guess, so what is your starting point for this? Do you have a metabolic model, or like how do you envisage you're going to go towards producing these materials?
So we're starting from some model materials which people have been studying for a while, so that we do have some understanding of their behaviors. And this product is more of a synthetic biology product. So our first goal or role is to develop synthetic biology tools so that we can synthesize these materials, and we can have tools to allow us to control the structure and some of the properties of these materials. And once we have success from that, then we'll move forward towards addressing other problems more in terms of how we can make significant amount and how we can address other issues from the material aspects.
Okay, so it's the first step is working on the tools to really engineer cyanobacteria in a high throughput way, I guess.
Correct. This is one of our aim.
Yeah. So, Eugene, what's your, if you're just starting, do you have an idea of what your role will be as you start your PhD? Are you a molecular biologist, or are you more in the kind of computing side of things or anything?
So, so for the few weeks that I've been working on this project, I've kind of been preparing like some of the monomers that serve as like a basis for some of the materials that we want to produce. I've also been working on, I guess, designing some of these monomers so that they're more easily, I guess, like detectable or like easy to visualize. And hopefully it'll serve as a foundation for when we move kind of like downstream of the project, where we're actually getting to the actual synthesis of these more complex materials.
Okay. So how do you, what will be the first step then? I mean, do you have like a first molecule you intend to work on?
Yeah, so we have an idea of, I guess, like the monomer that we want to use, but right now we're still kind of working on how these monomers will be able to polymerize with each other, just for, yeah, for the production of the more complex materials. We're also working on ways to, like, because we need to be able to detect these monomers to see how they behave in both like in vitro and in vivo systems. We want to look at ways we can design these monomers in terms of like the structure so that we can get the more complex materials.
Right. So how do you do your analysis? How will you be doing it?
So right now these are proteins we're working with. So and they're very small. I mean, obviously you can't see them with the naked eye. So we want to, I guess, like your classic detection of proteins, whether they be on, I guess, like a blot or just other visualization techniques using other types of tags. Yeah, we're still learning a lot about it right now. So yeah, we're still in like a work in progress.
Okay. Right. So can you tell me a little bit about why? So yeah, so these molecules aren't made in cyanobacteria already. So I presume they're not already made by cyanobacteria. If that is the case, what are you going to take enzymes from other organisms, or are you going to try and synthesize a new pathway de novo? That's something going on.
Right. So the materials that we are looking for, or we are studying, are mostly protein-based materials. So they are not like materials that are polymerized from small molecule monomers. So in that case, we have a much easier way on their synthesis, which basically just control the synthesis of these proteins. However, they are unique proteins. They're not like water-soluble, those sort of common proteins, because they do exhibit material properties. So they have their own characters you have to deal with. So there are lots of concerns we have on that aspect. So in a way, we're not going to develop new metabolic pathways to the synthesis of these materials, and bigger of a focus will be how you can control a better synthesis of these material protein monomers, and then we'll be able to control their assembly to a material exhibiting the exact properties as NASA wants.
Oh, that's really interesting. So, well, yeah. So does that make it easier than traditional metabolic engineering approaches then, if you're working with proteins?
Yeah, well, it's in both ways. In terms of their direct synthesis, you can think, well, it should be easier because you just make one protein instead of a series of enzymes. But on the other hand, they are way much harder proteins to work than any enzyme probably involved with hydrolyte biosynthesis.
Cool. So do you, I mean, I have a limited experience overexpressing proteins in E. coli, and you end up with these, obviously, big inclusion bodies is a large problem. Is this something that you think you might encounter with cyanos?
Yes, correct. So that is the red problem that we're facing now. And particularly because, as I said, these proteins are more like material proteins. You do not expect them to be soluble at high concentration. So they do try to come out. And we're trying to, one of the tools that we're trying to develop is how to make them be able to assemble even though they are not highly soluble, and how to control, how to engineer the host so that cyanobacteria can tolerate these proteins and cope with them and allow the proper assembly of these protein monomers, even when they are not quite soluble.
Okay. So has anyone actually done anything like this before, or is this completely novel?
Well, I guess Chris has some experience to work with these monomers in E. coli, and so we know something about them. We're still learning more about the behavior and of these, both the organisms, the host, and also the behavior of the protein monomers and later the polymers.
Okay, so Chris, can you tell us a little bit how your work's gone so far? Like what kind of problems you've run into or how you've managed to address these?
Well, yeah, it's fundamental right now, but we've been able to make some materials. We haven't really characterized the properties yet, though, and that's the next step.
So what kind of material? Like, do you have to break the cells to get it out, or is it excreted into solution, or?
Yeah, well, we can do either. We hope maybe someday we'll be able to actually make the materials outside of the cell, but right now, yeah, we make it in the cell and then we lyse the cells, and then, like Eugene was saying, right now it's pretty basic protein sort of methods like Western blotting and Page, and maybe also agarose gel electrophoresis for large assemblies.
Okay, that's cool. Right, let me see. Yeah, okay, so I mean, a little bit more about cyanobacteria in general. In terms of how is the tool set already for using? So for example, other people who are thinking of using cyanos for doing their own research, is there much out there in terms of, is there well-established like Golden Gate systems or anything like this for cloning?
Right. So people have been studying cyanobacteria for several decades, so there are definitely a significant amount of systems biology knowledge there. However, in terms of synthetic biology, I think it's really only been there for maybe a decade or even less. And it behaves quite differently in many aspects with other model organisms. For example, they grow way much slower than regular bacteria like E. coli. So students have to have lots of patience to work with them. And the genetic tools for you to modify their genomes is not quite the same as E. coli because there are multiple copies of the genomes which bring actual problems to us you have to deal with. And many of the tools that were originally developed for other bacteria, they do not work in cyanobacteria that well. And even right now, people are suffering from the lack of multiple orthogonal inducible systems that you cannot control the expression of multiple genes quite as a way as you can do it in E. coli or other microorganisms. So all these tools that are strongly needed for the success of this product. At the same time, we are through the other product and also since we started this product, we have been making some of the tools that would allow us to modify its genomes and control gene expressions in a much easier and much better way than what people can do in the past. So I think we're getting there.
So what kind of things are you using to develop these inducible expression systems?
So we're not particularly developing the inducible systems. There are several groups has reported fairly recently on those. For example, people a few years ago just borrowed those like inducible system or the Rabinow's inducible system into synyctids or other Syncococcus, different cyanobacteria species, and they found right away they're either very leaky or they're not inducible or they do not have the correct dynamic range at what you would be able to obtain from other organisms. But ever since then, there has been some knowledge that how you can edit the sequence of a promoter or certain elements that would allow it to work a little better than before, but still their behaviors is way much worse if you compare to how they behaved in E. coli. And on the other hand, which probably is more interesting to us, is basically to very quickly to modify the genomes of a cyanobacteria at multiple locations, so that we can will be able to allow us to control expression of multiple of those protein monomers, and in a very quick way. So we have been developing some of the tools in some recent knowledge to do such type of thing, and at the same time will be how you can regulate the expression pattern of these genes, because a uniqueness of cyanobacteria is that many of those species, because they have photosynthesis, so they have a circadian rhythm associated with their growth, with their metabolism, and with their gene expression as well, which has both pros and cons, because it brings some unique properties that you can utilize, but at the same time it has some problems that you have to face. That would require a unique way for people to develop some special tools so that we can utilize such a feature to control the expression of multiple genes. That's another tool that we have been working on mostly on the other product for a while.
Okay. I mean, I know that you can perform homologous recombination cyanos, right?
Oh, correct. So in terms of genome modification, so homologous recombination has been used in the past for quite a few decades. However, one problem, as I said, is cyanobacteria has multiple copies of genomes. So unlike E. coli or other organisms, then you just as long as you see the colony start to grow, then you are good. But on there, you see a colony, maybe it means you only have one or a few copies of genomes, have your target gene integrated so that they can grow. But you still have the wild-type genes there. And over time, because of the burden from expression of heterologous genes, and the wild-type gene will take over and will, so still using homologous recombination to replace your target gene. And actually, this process could occur in a very short of period, short of time, saying let's say a few weeks if you let the bug to continue to grow. So then, which means very quickly you lose the gene that you integrated. In a way, you have to create a stable construct by means of fully segregation, which usually takes several weeks, or sometimes if you're working with a hard gene, could be months. So we're trying to solve this problem by having a system that would allow us to very quickly fully integrate the whole genomes, all the copies with the target genes. And also at the same time, we wish that we can do multiple targets in the same batch as well.
So do you mind detail about that? Is that a genome editing technology, or is this something you want to keep secret?
Well, so yeah, I can talk a little bit about that. I think there are plenty of tools that people recently developed. For example, CRISPR is something that everybody is using in the field of synthetic biology, which turned out to work very well in cyanobacteria as well. So we're trying to utilize that system to see whether that can help us with solving some of these problems.
So have you had plenty of results from it already? Does it look successful?
Right. So right now we do find a system that would allow us to engineer the genomes fairly efficiently.
Okay, that's cool. So do you envisage any other, so beyond the protein material production, do you envisage any other use of the tools you're developing then?
Yes, of course. I think, for example, this genome modifying tools, I believe that can be used in other field of metabolic engineering or other field of synthetic biology as well. And at the same time, the gene expression tools that we develop will be useful to control other pathways, or even to use to study the biology or biochemistry of cyanobacteria, so that people can use that to perturb or manipulate a special gene of interest to look at how their circadian rhythm would affect their metabolism or their biochemistry. I do think these tools will find applications in other fields as well.
Cool. Well, great. Oh yeah, I have a few quick questions, but this has been great. So with the tools you're producing, do you envisage making them open source, or is something is IP something that is involved in your project?
The open source? Yeah, I mean, we're all fans of open source, at least I am, but you know, I guess science is still a competitive endeavor. So at this stage, we do have things that we have to keep to ourselves, but ultimately, everything that we produce, we will publish and disseminate. So yeah, I think we're all in favor of that.
Cool. Does your university have a well-developed setup for do they encourage startups and things like this?
Yes, correct. So Washington University does encourage us to pursue that route.
Okay, cool. Well, thank you very much. It's been really enjoyable. And did you want to say, you're looking for someone to join your NASA project? Is that correct?
All right. Great. So I think this might be a good opportunity for us to advertise this product. So I think we have been talking about what this product is about, and at the same time, we also have other funded products to on the similar direction, but using other organisms or other platforms to for the production of renewable high-performance biomaterials. So currently, we're looking for a postdoc with particularly biomaterial experience or with protein engineering background. That if you're interested in this topic and if you're interested in moving to St. Louis, so please contact me.
Okay, cool. Thank you very much. Well, it's been nice speaking to you all.
All right, thank you so much.
See ya.
Bye-bye.